So hello students, welcome back to your own channel Vedantu NEET English. This is Basratso, your biology master teacher. Good evening each and every one of you. Welcome to the class. Now students, this class is actually going to be very amazing, and it is not any other one shot on the YouTube. Why? Because today I'll be showing you the bigger picture. When I say the bigger picture, I mean visual representation. 3D models of how exactly the photosynthesis takes place. Not just the photosynthesis, even the chloroplast, even the anatomy of leaf, I will be showing you in a 3D image. Do you want to see a small glimpse of it, students? Do you want to see a small glimpse? Right here, students. I'll show you a small glimpse. This is how it's going to be, students. This here, I believe all of you can see this. This is a typical leaf, and here we go. Do you see this? This right here is the upper epidermis. This right here is the lower epidermis. Between the two epidermises, there is a small hole called here where the CO2 enters through the stomata. It enters, and these are the palisade mesophils. Palisade mesophils right here. Palisade mesophil right here. And if you see right here, this is the entire vascular bundle where the entry and exit of the phloem takes place, where the sugar is being transported. This is how it's going to be. This is how it is going to be today's class, because I will be showing you exactly where in the entire leaf non-cyclic photophosphorylation happens, where exactly C3 cycles happens, where exactly C4 cycle occurs in a plant. So students, are you excited for today's class? Are you excited? Tell me in the chat. Quickly tell me in the chat. Are you excited for today's class? On that note, students, on that note, let me tell you something. Back in the days, in the early 90s, people used to think that plants grow by eating soil. Yes, you heard me right. Back in the day, people used to think that plants are actually growing, they're increasing their biomass by eating the soil. For many years, this was a theory that we have the plants here, they will start eating the soil and they will grow in size. But that is not the case. All of you know that is not the case. This theory was eventually disproved by Helmholtz, which is a scientist. And why am I sharing this? Because it is a nice information, right? It is not given you a syllabus, but yeah. Similarly, the term photosynthesis was given by your Barnes. He termed the coin photosyntax, like a small tax, okay, photosyntax, but the word photosynthesis became more popular eventually. It became more popular eventually, and that is photosynthesis. But what exactly is the meaning of photosynthesis? I'll give you a small, let's play a small game, student. Let's play a small game. Tell me what is your favorite food in the chat quickly. What do you want to cook? Quickly tell me in the chat, what is your favorite food item? Let's cook it. For example, if you're a chef. And you're trying to make, let's take some chicken biryani. Okay, chicken biryani. You bring chicken, you bring rice, you bring vegetables. If you add vegetables, that is. You bring oil, you bring spices, you bring every single thing, and you add entire thing in a pot. So, to make the biryani, did you bring a lot of ingredients? Yes, to cook you need ingredients. Similarly, students, if you want to do, you go for shopping. You go shopping to find all these ingredients. But what do you think is the ingredients for plants, students? What do you think are the ingredients used by the plants for photosynthesis? Quickly in the chat, all of you. What are the ingredients which plants use to make the food? Like for biryani, I told you just now, we need rice, chicken, everything. If veg biryani, some vegetables, okay. But if you want to photosynthesis, the main ingredients are, let's take plants need CO2, plants need chlorophyll, plants need water, plants need sunlight. All of you agree with me? Let's write down, students. Let's write down the ingredients which plants require. For example, let's take a typical plant cell. Here. This is a typical plant cell. In this typical plant cell, plants need CO2. Okay, plants need CO2. Okay, CO2 is entering. Now students, this CO2 Plants need sunlight. Plants need sunlight. Now, when there is sunlight, this CO2 is getting converted to a structure. It is a sugar. Basically, this CO2 is getting converted to C6H12O6. What is this? This right here is the glucose. Right here is the glucose. So CO2 is getting converted to glucose. Now students, can you tell me the difference between this and this? See, here I have carbon, oxygen. What is missing there? What is the missing element? The missing element is water. So do we add water as an ingredient? Yes. We also add the H2O. So what happens here? This hydrogen is donated. This hydrogen is donated in the formation of glucose. If this hydrogen is donated to make the glucose, this oxygen is released as a byproduct. And that is the simple process. That is the simple process of photosynthesis, where plants use light energy. Plants are using light energy to synthesize food, to produce food. That is called as photosynthesis. Beautiful, no? Beautiful process, right? This entire beautiful process, students, let me show you. This entire beautiful process occurs. This entire beautiful process takes place in a structure. Can anyone tell me, students? This entire beautiful process takes place in a structure called as chloroplast. Why am I not able to zoom it? 1 second, students. This entire thing takes place in all of you are able to see this? Entire thing takes place in a structure called as chloroplast. Now we'll come back to this chloroplast eventually. Where I'll be teaching you what exactly is this. What is this thylakoid? What is this entire structure? Is it single membrane, double membrane? What is this connecting link? What is the matrix called as? So we will get back to this entire thing. Right? We will get back to this entire thing, chloroplast, eventually. But before that, let me tell you a basic things about photosynthesis. Okay. Now students, the first basic thing which here you need to understand here is, from here to here, can I tell there was addition of hydrogen? There was addition of H plus. Yes, addition of H plus. Now this addition of H plus is called as, what do you call it, students? Addition of H plus, is it called as oxidation or is it called as reduction? Students, quickly tell me in the chat. This addition of hydrogen is called as a reduction process. Reduction. So remember it. This entire photosynthesis is a reduction process. Now in this entire reaction, there is absorption of sunlight. So can I call it as endothermic reaction? Endothermic reaction. Yes or no? It is a complete endothermic reaction. Now in this entire process of photosynthesis, inorganic substances are converted to organic substances. Inorganic to organic substances. Now one more thing, students, endothermic, inorganic to organic, reduction process. Now, do you think this process of photosynthesis, does it require enzymes? The answer is yes. This entire reaction is also enzyme mediated. It is also enzyme mediated. Okay. Now, if you remember the basic concept of anabolism and catabolism, here I am forming organic molecule. From simple substances, I am making a complex substance. Do I call it catabolism or anabolism? Quickly in the chat. I will call it anabolic process. Anabolic. Because I am making complex substance from a simple substance. So what is photosynthesis? Photosynthesis is a process. It is a enzyme mediated process in which inorganic to organic conversion has happened, It is an endothermic reaction. It is a reduction process in which glucose is produced, and oxygen is released as a byproduct. So I believe right now all the students of mine has a clear cut idea about what exactly is photosynthesis. If this definition is clear to all of you, quickly green hearts in the chat for the bot army. Quickly, green hearts in the chat for the bot army in the go. All of you, quickly. Now, this is the entire process of photosynthesis. All of you know there is carbon dioxide plus water, sunlight, chlorophyll, glucose plus oxygen. Reactants, products. This glucose is taken by us eventually. It is taken by us eventually. Now you might ask me, sir, what is the importance? Why do we even need photosynthesis? Basic thing. Students, tell me. Can all of you, let's take object, let's take Disha, let's take Susmita, let's take Sneha, let's take Pratibha. Can any one of you even survive without food for at least 10 days? No. Now imagine a life without food itself. Not possible. Because every single food which we eat right now is directly or indirectly coming from plants. So technically speaking, we cannot sustain our life without plants. Right? Further, there is primary source of food on earth responsible for the release of oxygen. Obviously, oxygen is a byproduct here which we take in. So we cannot survive without plants. So save plants. Okay. Now students, with that basic introduction, now we go to a concept called as early experiments, where many scientists, where you have to give namaskara now, namaskara, okay, many scientists gave contribution in the advancement of understanding the photosynthesis and the entire process of photosynthesis. Let's understand the early experiments now. Shall we start? Now the rule is, after every single experiment, if you understand it, start putting hearts in the chat if it is clear to you. Can we start? Can we start, students? Yes. Now, the first experiment, before I show the first experiment, I believe all of you have seen such plants in real life. Such plants in real life you might have seen in your locality. You might even have it at your house also. There's a very high chance that, yes sir, I see this plant every single day. Now students, such plants are called as variegated leaf. Plants which have such a leaf is called as variegated leaf. The meaning of variegated means variety of leaf they have, or in a particular leaf, a particular section will have more chlorophyll compared to the other, and that leads us to the first experiment called as variegated leaf experiment. First of all, let me tell you right now what is the advantage or what is the outcome. The outcome of this experiment is, this experiment proves that there is a requirement of chlorophyll. Without chlorophyll, no photosynthesis, like no fun in the party basically. So without chlorophyll, there is no photosynthesis. Okay, understand. Now what are we doing? We are taking a variegated leaf and we are taking this variegated leaf and we are keeping it in sunlight. Now after some time, we are adding iodine to it, and we notice that wherever we have the dark green regions in those specific locations, we have violet black color or blue black color, and in the yellow region or light green region there is no color change. It tells you what? It tells you that in the dark green regions there is a pigment here. Can anyone tell me what is the pigment? The pigment which I'm talking about, the elixir of life. The pigment which I'm talking about here is the chlorophyll. So in the dark green regions, since chlorophyll is present, hence photosynthesis occurs, and hence the product of photosynthesis, that is starch. The starch is reacting with iodine, and that starch and iodine is giving the blue black color. And wherever there is no iodine, we generally get no coloration. That is all is the experiment, variegated leaf experiment, right? Chlorophyll is necessary for photosynthesis. No chlorophyll, no color change. Chlorophyll, color change. Simple as it gets. Simple as it gets. That's all. Next experiment is called as Moll's half-leaf experiment. Very simple again. Very simple. Ultimate focus. Listen to me very carefully. Listen to me very carefully. Okay. Moll's half-leaf experiment is very simple. Here we need to add some agent called as KOH. KOH. Understand? For example, students, in this particular jar, let's take a jar here. In this jar, this portion of leaf is kept under presence of KOH. KOH. Potassium hydroxide. Potassium hydroxide. Right? Potassium hydroxide. The above part is free in there. Free in the air. Now, what exactly happens here is this region which is under KOH. This KOH is a kala. Okay, it's not criminal. It is criminal. It is criminal. I'm telling you, criminal it is. Okay. This KOH completely absorbs all the CO2 present in the environment. So what is happening here? The entire CO2 is being consumed. So no KOH at all. No KOH. So and there is no CO2 at all. No CO2 at all. In the above region, CO2 is present. Carbon dioxide is present in the above region. It's present. So what happens? In the starting I told you, CO2 gets converted to glucose. If there is no CO2 at all here, do you even see starch formation here, students? Do you find any starch here? Sir, what are you telling? No CO2, no starch formation. Above part, starch formation is happening. So this experiment shows that. CO2 is essential for photosynthesis. That is all is experiment. That is all is the experiment. Now students, I want everyone to quickly smash the like button. Quickly throw some chappal and smash that like button. Okay, whatever you want to do. That is Moll's half-leaf experiment. Now we have the famous bell jar experiment. Very simple again, very simple. Okay, little bit sad story also. If you like mouse, it is a little sad story. Okay, what happens? Understand. There is a bell. This is called as a bell jar. Now there is a mouse and a candle inside a bell jar, and let's say after some time, let's take around 1 hour. After 1 hour, let's take 11 hours. Okay, after considerable amount of time, okay, the mouse is RIP all of you in the chat, students. RIP for the mouse, gave its life for the science. So this mouse undergoes RIP. It becomes RIP. Okay, and this candle also becomes extinguished. Candle also is going poof. So this shows that something was present in the air. Something, we don't know still. Something was present in the air which got consumed by the burning of candle and the respiration by the rat. So the rat is respiring, candle is burning. Because of these two conditions, entire oxygen, spoiler alert, it's a spoiler, entire oxygen is being consumed by the bell jar, inside the bell jar. Now in the second case, this is case 1, and this is case 2. In the case 2, what are we doing? In the case 2, we are keeping a mint plant on the inside. There's a mint plant on the inside, case 2. Okay. Now, because in the case 2, every single time the oxygen is being consumed, the oxygen is replenished. It is being replaced by the mint plant. Yes or no? So, even after 10 hours, the mouse is alive, candle also is burning very well. So, Joseph Priestley experiment, our Uncle Joseph, okay. Hypothesis, plants are restoring. So, okay, plants restore to the air whatever breathing of animals and burning of candle removed. So, oxygen was being removed from the environment and it is replaced by our amazing plants. Okay, it is replaced by the amazing plants. Now students, does it happen in your class that there are some people in your class who will copy your homework and they'll get more marks, free marks they'll get? Does it happen? Same thing happens here also. Okay, same but different. Small difference is there. Understand? There is a scientist called as Jan Ingenhousz. He copied the entire experimental setup of Joseph Priestley with a minor change. Now, what is this minor change here? The minor change is very simple. He took the experiment in the sunlight and in the darkness. In sunlight and in darkness. He noticed that in the sunlight there were bubble formation, and in the night time no bubble formation. What does it tell you? What does it tell you? Okay, what does it tell you? It tells you that, students, in sunlight there is release of oxygen, in the night time no oxygen. So what is the significance of this oxygen? The significance of this oxygen is that all of us know oxygen is a byproduct of photosynthesis. So can I tell, in the sunlight photosynthesis is happening, in the night time photosynthesis is not happening? Simple, no? Very simple. He saw the bubbles and he gave the experiment. Newton saw the apple, he gave the explanation. Jan Engineer saw the bubbles and he gave an explanation. Smart, no? Very smart they are. They see small things, observe the small things and tell some extra information for all of you. Okay. So it tells you that sunlight is very important for photosynthesis. Okay. Then we have Julius von Sachs. Julius von Sachs experiment. Julius von Sachs experiment told us very simple things. Very simple things. That is, provided evidence for production of glucose by plants. He told us plants are giving you glucose, be happy in life. Chlorophyll is located inside the chloroplast. Chlorophyll is a pigment. It is present inside chloroplast. Leaves synthesize glucose, which is later stored as starch. So glucose is stored as starch. That is all. 3 main points, done. Students, if you are able to follow through, green hearts in the chat. Every single experiment. Okay, every single experiment. Okay. Now, that is Julius von Sachs. Now, there is one more experiment called as T.W. Engelmann experiment. T.W. Engelmann experiment. Very important. If you don't listen to me right now, there is a very high chance that you will lose these marks. So I want everyone to give ultimate attention for the next exactly 5 minutes. Let's go. Can we start? Can we start, students? Can we start now? Can we start? All of you? T.W. Engelmann experiment. Ultimate focus now. Ultimate focus. Listen to me very carefully. Okay. What did he do? So all of you know there is something called as white light. And white light, when it passes through a prism, there is scattering of light. Basic concept, all of you know that white light will scatter under a prism. And we get a waveguide. We get the entire spectrum of lights. We get entire spectrum of lights is obtained here. Okay. Now, students, I will show you one small algae which he used. One algae. This algae is called as cladophora. This is the algae. Beautiful, right? Beautiful algae. Okay. So he used the algae called as cladophora algae. Understand? Very important concept. Now, this is the light coming out here. This is the light coming out here. Just focus. Red light as well as the blue light coming out here. Okay. Now say this. Here I am placing a container. Here I am placing a container. Okay. Now in this container, I am adding the algae which I showed you, the cladophora algae. Okay. Here I am showing not the DNA, it's an algae. Okay, it's an algae. Cladophora, it's a branched algae. Cladophora is a branched algae. Okay, I'm growing the algae here. Now within this jar, I am also adding a bacteria. Specifically speaking, a aerobic bacteria. Listen to me very carefully now. In this jar, I am adding the aerobic bacteria. Aerobic bacteria right here. Aerobic bacteria. Now students, this aerobic bacteria loves air. Aerobic bacteria loves oxygen, to be very specific. It loves oxygen. So all of you, will give you one simple. There is Hari Chan. Okay, let's Hari Chan. Let's say I am telling you tomorrow free chocolates for everyone. If you come to my class tomorrow, free chocolate for everyone. Will all of you come to my desk and like, sir, one chocolate, free chocolate one. I'll give you dairy milk silk for everyone. You'll come and take from me. Yes or no? You'll come and take. You'll be very happy. So if I tell this bacteria at a very specific locations, on this cladophora, there is chocolate. Will they go? They will go. Same thing is happening here. I am telling this cladophora, I'm telling this bacteria that, aerobic bacteria that, on this cladophora, at a certain region there is more oxygen. They will start forming a gang here for the free oxygen. Can you see all of you? One gang formation happened here. Similarly, there's one more gang formation here also. One more gang formation is happening here also. Okay, gang formation is happening here. Now you might ask me, why is the gang formation happening there? Why? Because, listen to me very carefully, this is the concept here, this is the concept. At these locations right here, I told you there is more oxygen. There is more oxygen. That is why aerobic bacteria is forming a flock here. There is more oxygen at this point right here. That's why the aerobic bacteria is forming a gang in these two places. It's forming. Now, why do you think there is more bacteria, more CO2 here? So more O2 here? Because in these two regions, if I tell there is more oxygen, can I tell in these two regions there is more photosynthesis occurring? Because oxygen is a byproduct of photosynthesis. So can I tell in these two regions there is more photosynthesis occurring? Photosynthesis is more. Photosynthesis is more here. Photosynthesis is more here. Photosynthesis is more in these two locations. It is more. Now, what do I call this? Two regions. What do I call these regions? These two regions are the red region and the blue region. Red and blue regions. So, what do I understand by this? Red and blue is the favorite color of the plant. Is red and blue the favorite color? Absolutely. That is, whenever there is red light, whenever there is blue light, plants love it. They go mad, they go mental. Like, oh, red light. That's how they act. Okay. So plants love red light and blue light because in these two lights can be absorbed better by the plants. If there is more absorption of light, there is more photosynthesis happening here. And that is the theory behind T.W. Engelmann. That is, in the red region and the blue region, there is more absorption. There is more absorption of light as well as more photosynthesis in these two regions. More photosynthesis in these two regions. So, with the help of aerobic bacteria, we were able to confirm that in these two regions there is more oxygen. See here. Use prism light split into the spectral components. Light was used to illuminate green algae cladophora placed in a suspension of aerobic bacteria. And we noticed that in the blue region and the red region, in these two regions, in blue region and the red region of the spectrum of light, there is more absorption. There is more absorption of light. There is more aerobic bacteria here. There is more photosynthesis also happening more. Beautiful, right? Beautiful, right? Because it is getting absorbed better. Similarly, students, I was going to save it for later, but let me tell you now only. Do you know why? Plants look green color? How many of you know that? Tell me. How many of you know why plants look green color? Why plants look green color? Green color, quickly. Why the leaf is green? Tell me. Why, students? Why? You might tell me because of chlorophyll. Because of chlorophyll. It is correct. It is correct. Technically, it is correct. But the entire, out of the entire spectrum, out of the entire visible spectrum, your chlorophyll is actually reflecting the light. Which one? Green light is reflected more. The green light is reflected more, and henceforth we are able to see with the help of rods and cones the green light more. and the red light, blue light is getting absorbed better by the plants. Beautiful concept. You learn this in physics more and zoology more, that whenever there is spectrum of light, all lights are falling here, the green light is being reflected out, and we are able to see the green color because it is being reflected off and it is not absorbed by the plants. So if I tell you red light and blue light is the favorite light, which is the most light which they don't like, then students tell me, which is the light which plants don't like? Tell me quickly, tell me in the chat now. Which is the color of the light which plants don't like? Green color. Green color, they reflect it off. Green color they don't like, and that is why we see the green color also. Okay, beautiful concept also. Someday I'll teach you more on that. Okay, now students, all of you understood TW Engelmann experiment? That is, aerobic bacteria detects the presence of O2. Yes sir. Formation of O2 highest in the red and blue light. Yes sir. Blue and red light wavelengths optimum for photosynthesis. Three main points for your TW Engelmann experiment. TW Engelmann experiment. Done. Okay, so till now we have understood that CO2 is very important. Mole's half-life experiment. We know water is very important. Yes, that I'll teach you now. We know sunlight is more important. That is a engine house experiment. We know chlorophyll is important. We know what is glucose. We know oxygen is being evolved here. So the entire puzzle is being solved little by bit. Little by bit the puzzle is being solved right now. Puzzle is being solved. Okay. Now the only thing which is missing is, why is the water required? Like for example, we tell what are the plants? What are the plants? But why? Why do you think plants need water? Think all of you. Use your brain cells. All of you use your brain cells and tell me, why do you think plants even require water at all? Why can't they just, are they really that thirsty? Is it possible that they're really thirsty? Tell me. Do you think they're really thirsty at this point that they're like, oh, I need water, what is the need of water there? Anyone? What is the need of water? Think. I'll give you 2 minutes. Okay. Now the last scientist is called as Cornelis van Niel experiment. Okay. He was a microbiologist. His main work was on green and purple sulfur bacteria. Main work. Okay. He noticed that students, whenever oxidation of water happens, whenever there is oxidation of water, there is a liberation of O2. There is liberation of O2 is happening. Okay. And water provides hydrogen in order to reduce CO2 into carbohydrates. First slide. Let's go back there. Everything is connected. Everything is connected. We learned here, right? We learned here in the first slide that CO2 is reduced to glucose. Why? Because the hydrogen was provided here. Hydrogen was given. So the importance of hydrogen, the importance of water is what? Electron donor. I'll repeat again. The importance of hydrogen, the importance of water is that it is a source of hydrogen and electron donor. Hydrogen and electron donor. So if I ask you a question, who is the electron donor? What is your answer? In the process of photosynthesis, the electron donor or the hydrogen donor is the water. Now, how did we know that? Right? Oxygen evolved from green plants come from the water and not from CO2. This is very important. All of you should be knowing that in this entire reaction, in the entire reaction of photosynthesis, CO2 plus H2O will give you C6H12O6 plus oxygen, O2. This O2 here is coming from the water. So this hydrogen here is splitting, water is splitting up. Hydrogen is given here to CO2 to get converted to glucose. This oxygen is released as a byproduct. Do not be confused. It is not coming from CO2. This oxygen is not coming. It is that this oxygen is being released. I hope that is very understanding for you right now. I hope that confusion is clear to all my students. The importance of water is hydrogen donor, and the oxygen which we get here, it is from water, not from CO2, not from CO2. How do we know that? How do we know that? Because students, in the case of purple and green sulfur bacteria, there is no water there. The hydrogen donor, there is H2S. The hydrogen donor, there is H2S. For example, here we have H2S. H2S here. So in the case of bacteria, how we have O2 is being evolved, right? O2 is being evolved in plants. In bacteria, sulfur is being evolved. Sulfur is being evolved there. Beautiful concept. Okay, sulfur is being evolved there. Okay, sulfur is being evolved there. From here, sulfur, this hydrogen is donated here, and the sulfur is released as a byproduct. Like how oxygen is released, right? Sulfur is released here. And with that, students, with that, we come to the site of photosynthesis. Site of photosynthesis. Where does the photosynthesis actually occurs? Where does it happen? Where does it happen? Ready, all of you? Ready? Ready, students, to learn? Ready, 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 ready? Yes? Now, students, try to imagine you are in a dense forest. Try to imagine you are in a dense forest like this. And in this dense forest, you are looking at leaves. You're like, so such beautiful leaves are there. But what exactly is present inside one leaf? How do you notice that? How do you notice that? See this. If you notice inside one leaf, if you go, right, if you open the leaf and if you look inside what is present, you can see thousands of, on the surface, stomata. Thousands of stomata can you see. Now if you go inside one stomata, right, let's travel inside the stomata now. If you go inside the stomata, you can see the beautiful world. You can see the beautiful world of plants, that is the cells inside, the internal structure of the plants, internal structure. Now there are a lot of cells here. There are a lot of cells. You can see lots and lots of cells, right? Lots and lots of cells are there, right? But I want you to focus on one specific organelle here. That is, if you look inside a cell right here, and if you go looking around everywhere, you will find a very specific organelle here called as chloroplast. Students, chloroplast. Okay, one specific organ called as chloroplast right here. This right here is the chloroplast. And if you look into the chloroplast right now, if you look in the chloroplast, you will find the actual location. You will find the actual location where the entire process of light harvesting is happening. And this right here is a thylakoid. Can you see? On top of thylakoid, do you see all these tiny structures? They are called as light harvesting complexes. They are called as light harvesting complex, which I'll be teaching you in some time. Light harvesting complexes, many are there. Many light harvesting complexes are there. Okay, many different light harvesting complexes are there, such complexes. Okay, so that is the exact location. That is the exact location where the photosynthesis occurs. Now, students, can we quickly draw the diagram once for our satisfaction? Can we draw, students, all of you? Can we start? Yes. Now, for example, this is a typical chloroplast. This is a typical chloroplast. Let's take green color. Okay. Now, students, a typical chloroplast which you saw right now, is technically a double membrane structure. It is a double membrane structure. It has an outer layer and inner layer. Can all of you see this? It has an outer layer, outer membrane as well as the inner membrane. Outer membrane and inner membrane. Two different types of membranes are there. Outer membrane, inner membrane. Now, if you look inside the membranes, there is a coin-shaped structure. We saw in the video, right? Coin-shaped structures everywhere. Coin-shaped structures everywhere. This individual, student, this individual coin-shaped structure, individual is called as thylakoid. It is called as thylakoid. Then what do I call this entire stack? This entire stack, students, this entire stack is called as grana or granum. It's called as grana. Okay. Now, students, understand very important concept. Individual thylakoid are connected to each other. They're holding hands. This connection is there. Every single thylakoid or the grana is connected to each other, and that individual connection is called as stromal lamellae. It is called as what? Or stromal lemma. Okay, stromal lemma. Okay, that is stromal lemma. Now, students, this entire thing has matrix. Do you see? This is the matrix. This entire thing has a matrix. The matrix is called as stroma. It is called as stroma. Now, listen to me very carefully. On top of this, on top of the thylakoids, there are two round structures here. Not two, there are two main structures. And these structures right here, looks like a caterpillar, no? Looks like a caterpillar. Okay, two round structures here are basically the photosystem 1 and photosystem 2. Not PlayStation, photosystem 1 and photosystem 2. Both are present on the thylakoid. Very important concept. Both are present on the thylakoid. While if you look at the stromal lemma, on top of stromal lemma, we only have the PS1. Only photosystem 1 is present here. So photosystem 2 and 1 both are present on the thylakoid. On the stromal lemma, only and only PS1 is present. 1 is present. Okay. Now, apart from that, students, in the stroma, the dark reaction happens. Dark magic, basically. Dark reaction takes place here. And on the stromal lemma, grana, thylakoid, light reaction takes place here. Now, what is light reaction? What is dark reaction? Do not have to worry. I'll be teaching you the entire thing in today's class itself. One shot. Okay, one shot this is. Okay, so this is the entire diagram. Let's see this one more time in the here. I hope I'll be able to. 1 second, students. 1 second. Students, is it visible right now to all of you? I hope it is visible. See, this is the entire chloroplast. This is the outer membrane and inner membrane. Now, if you zoom inside, right? If you zoom in, let's take this is the inner membrane. This is the entire thing is called as the inner membrane. Here we have the thylakoids. Single thing is called as a thylakoid. This is the connecting link, lamellae, right? The connecting link. Then we have the stroma, the entire matrix called as the stroma, right? What is this structure? This is the, students, I hope it's visible right now. 1 second, let me just focus on this. See, this is the thylakoid. From outer thylakoid, inside the thylakoid part, that is called as lumen. The inner part is called as the lumen. Okay, inner part is called as the lumen. Apart from that, we can see some starch granules everywhere. We can see the ribosomes everywhere. Now, if you can remember, which type of ribosomes do you find here? In the case of chloroplast and mitochondria. We generally find 70S ribosomes. 70S. Okay, 70S. Okay. Now, this right here is the lipid molecules. Lipid molecules are also there scattered everywhere. Now, students, why this lumen is important to understand is, this lumen is very important to understand because when I'll be teaching you the chemiosmotic hypothesis, the majority of the reactions occurs in the lumen part. So that will be coming up. It will be coming up in the lumen. So lumen will have that. Lumen will have that. Okay. So I hope everyone is able to follow through now. Okay. Everyone is able to follow through. I hope everyone is understanding better today. This is the actual photosynthesis in 3D. Okay. Now students, all of you learned this 𝐂𝐤 𝐏𝐥𝐚𝐲. 𝐂𝐤 𝐏𝐿. I already told you this one. Why do they appear green? Because of the green light is being reflected to our eyes. Okay. Now students, but there are many other colors, right? Yes or no? There are so many different colors are there. Like there is not only green, there is yellow, there is red, there is so many different colors are there. So do you think plants only have one color or multiple color? Can anyone tell me? Do you think plants only have a single color or multiple colors in them? Are there any other pigments along with the chlorophyll? Yes, there are. There are actually multiple pigments and there is a method to separate them. Yes, there is. The method to separate all the pigments, the method is called as paper chromatography. It is called as paper chromatography. Okay. What are the pigments? So how do you extract it? All of you to be scientists? All of you ready to be scientists now? Scientists? Okay. So I want everyone to collect one leaf. Take a leaf and grind it nicely. Mortar and pestle, grind it. If you want to add a solvent also, acetate or something, some form of acetone, sorry, not acetate, acetone. Okay. And then you filter it. So you get something called as the extract. This is called as the extract. You take the extract and drop by drop add it on a filter paper. Drop by drop add it on a filter paper. Then take the filter paper, keep it in a beaker. Keep it in a beaker, and inside the beaker, there should be a solvent. There should be a solvent. Now students, understand. As the solvent will move up, because of the capillary action. All of you know what is capillary action? When you take a straw and you keep it in water, the water moves up through the straw without any force. That is called as capillary action. With the help of capillary action, the solvent will move up. When the solvent is moving upwards, it is also bringing the pigments along with it. And some pigments are more soluble, some pigments are less soluble. The pigments which are more soluble, carotenes, xanthophyll, chlorophyll A, chlorophyll B. More soluble, less soluble, and with the help of a simple method called as paper chromatography, we can extract the different pigments. So there is not only a single pigment, there are multiple different pigments present inside a leaf. Okay, present inside a leaf. Okay. Now, but I have to ask you now. I have to ask this question to all my students. What is a pigment? What is a pigment, students? Can anyone tell me what do you mean by a pigment? Do you think pigment is a chemical which gives color? Do you think that, students? Do you think pigment is a chemical which gives color? Yes or no? Tell me yes or no. Tell me. Pigment is a chemical which gives color. True, it gives color to us. But is that it? Is that it, students? No. What is pigment actually, students? Pigment is a chemical where electrons can get excited. That is a pigment. Understand? Understand, all of you. Okay. Pigment is a chemical. It is a chemical. It is a chemical that converts light energy into chemical energy. Understand all of you quickly. For example, here I have a pigment right here, let's say chlorophyll A. I have a pigment here. Let's take it as chlorophyll A. Okay. A particular wavelength of light is falling on it. A fixed wavelength is falling on it. Fixed wavelength is falling on it. Fixed wavelength is falling on this pigment. This is the pigment right here. A fixed wavelength is falling on this pigment. Now what will happen here? The electrons present in the outer orbital, the electrons which are present in the outer orbital gets excited here. All the electrons in the outer orbital gets excited. Electrons are getting excited. Now students, this electron right here, this electron has to come back to the ground state eventually. Basic chemistry that this particular electron has to come back to the ground state now. Now, when this electron is coming back to the ground state, it can undergo certain changes. For example, and listen to me. This electron can emit light. It can emit light energy, which we see in chemistry. Or it can emit energy. It can emit, let's say, heat as energy, heat. Or it can transfer the energy to another pigment. It can transfer the energy to another pigment. Or it can use the energy to do a function. It can use the energy to do a reaction. Now students, for our study in this chapter, we will be focusing on third point and fourth point, not on first and second. First and second you will basically learn in chemistry, but we will be focusing on transfer the energy to another pigment. Example is coming up. For example, the auxiliary pigments transfer the energy, they do. And energy to do some reaction. That is basically the chlorophyll A reaction center. Now what is reaction center? What is antenna pigment? What is light harvesting complex? What is a photosystem? You will learn exactly what is the meaning of these terms in the next 20 minutes. Can we start now? Students? Now, before we go to those higher concepts and this basic functions, I want everyone to understand two main points here. What is the meaning of action spectrum and what is absorption spectrum? Very simple, very easy. Okay, very simple it is. What is action spectrum and what is absorption spectrum? Okay, first of all, what is absorption spectrum? What is absorption spectrum? Now students, I told you, every single pigment is present in the plant. There is chlorophyll A, chlorophyll B, xanthophyll, carotenoids. Different pigments are present in the plant. Now there is a very high chance that, don't you think so, every single pigment gets absorbed at a different wavelength? Think. All of you will have a favorite biscuit packet. Tell me your favorite biscuit packet, students. For example, I love Oreo. Oreo is my favorite. Some of you will like, I like Parle-G. Some of you will be like, sir, I like Bourbon. I like the 50-50 biscuit. I like Jim Jam. So all of you have a different favorite biscuit. Yes. Similarly, every single pigment, it could be chlorophyll A, B, xanthophyll, carotenoids. They have their favorite wavelength at which they are getting absorbed. So wavelength at which a pigment is getting absorbed, that is called as absorption spectrum. The absorption spectrum of leaf pigment indicates the wavelength of light absorbed by each pigment. So each pigment is absorbing light at a particular wavelength. So what is the wavelength? Is a favorite wavelength. For example, if you look at this graph given in your NCERT also, chlorophyll A is getting absorbed here. It is getting absorbed here. Two different peaks for chlorophyll A. One is red, one is blue. Red and blue pigments. It's, for example, blue is for 400, red is around 700 here. Two different wavelengths, it is getting absorbed better. That is called as absorption spectrum. Now, what is action spectrum? The meaning of action means something is happening, some action is happening here. The action which are referring to here, is, for example, this is chlorophyll A. At this peak and this peak, light is getting absorbed. If light is getting absorbed, some action is happening. The action which we are referring to here is nothing but photosynthesis. So action spectrum tells you that at which wavelength photosynthesis is happening. Did we learn something very similar concept in the T.W. Engelmann experiment? In T.W. Engelmann experiment, students, in T.W. Engelmann experiment, we learned that at this red wavelength and blue wavelength, at these two regions, photosynthesis is happening more. It tells you basically this is actually the first action spectrum right here. So action spectrum tells you that at which wavelength photosynthesis is happening. That's all. Absorption spectrum, at which wavelength absorption is happening. Action spectrum, at which wavelength photosynthesis is happening. That's all. Now if you overlap these two, that is for chlorophyll A here, where is it? Here. Chlorophyll A, here. This one and this one. If you overlap these two graphs, you will end up getting this graph right here. And this graph clearly tells you that whenever there is absorption of light, there is also photosynthesis. So the black line represents photosynthesis and the light blue represents absorption. So wherever there is absorption, there is also photosynthesis. But students, one critical concept here, one small concept here. Okay. Here absorption is happening, photosynthesis is also happening. Same goes here. But from 500 to around 650, I can barely see any absorption. There's technically no absorption here. There is technically no absorption at all here. Then why is there photosynthesis happening still? Simple question is, chlorophyll A, this is for chlorophyll A. This is for absorption for chlorophyll A. Chlorophyll A is not getting absorbed at 500 to 650. Let's take 600 itself. 500 or 450. 450 or 470 to 600. There is no absorption of chlorophyll A. Even though there is no absorption at all, there is still photosynthesis. Why? Students, why? I'll give you a simple clue. Sometimes you forget your tiffin at home. You don't take your lunch box. Sometimes you don't take your lunch box to school and you go off to school. In the lunch break, do you simply keep quiet or do you eat your friend's food? Tell me in the chat quickly. Sometimes what happens is you forget your tiffin box in the house and in lunch break, do you simply keep quiet or do you eat your friend's food? Tell me, what do you do? You eat your friend's food. You're like, what is it, bro? What did you bring? What do you bring? You ask your friend. Yes or no? Same thing is happening here also. That is, even though chlorophyll A is not getting absorbed, at all, but there are friends are there, no? Who are the friends here? The friends which I'm talking about here is chlorophyll B, xanthophyll. Chlorophyll B, xanthophyll, carotenoids. These pigments, they're like, plus take my food. So these pigments are absorbing the light, they are giving the energy to chlorophyll A. These friends are giving the energy to chlorophyll A to do the reaction. So that is why even though chlorophyll A is not getting absorbed here, other pigments are helping out. They're absorbing energy, they're giving the energy to chlorophyll A to do the entire reaction. So friends are helping out here. This is for chlorophyll A. All of you understood? So we have, by that definition, we have two type of pigments. One is called as the chief pigment. Example is chlorophyll A, chief pigment. There is also accessory pigments. Friends, I told you, friends. Chlorophyll B, carotenoids, xanthophyll. Color is important. Blue green, yellow green, yellow yellow, yellow orange, yellow. Color is very important. Color also is very important. So chief pigment is the one which is like the ultimate boss, and he is the one who's sitting at the top. Every other pigment is giving the energy to the chlorophyll A. Let me show you a diagram, you'll understand better. Okay, you understand better right now. Now this is auxiliary pigment. Auxiliary pigments absorb light and transfer it to chlorophyll A. I'll give you this. This is chlorophyll A. Additional information. Chlorophyll B. There's small changes are there. Okay, small changes there. Understand, all of you now. Understand what exactly is happening here. Understand, all of you. Understand what is exactly is happening here. Okay, let's take chlorophyll A. Right now. This is chlorophyll A. 680 nanometer it is working. 680 nanometer chlorophyll A is working. At 680 nanometer, the ultimate boss. Here we have the underlings. All of you seen movies, right? In Pushpa or any KGF movie, anything. The ultimate main boss has a lot of people under him. He has a lot of people under him. Left hand, right hand, one left, one head, one tail. You see people, right? So on the top we have chlorophyll A working at 680 nanometer. Okay, 680 nanometer. At the same time, we have the carotenoids. Carotenoids which are working at, let's say, around 500 to 550. 500 to 550. Okay. Then we have xanthophyll. They are working around, let's say, 550 to 600. 550 to 600 is their absorption range. Is there. Okay. Then we have the top, that is chlorophyll B. Which chlorophyll A can trust more, brother. Okay. He is working, let's say, around 600 to 680. Now, students, when there is incident light, out of this entire light, 680 is absorbed by chlorophyll A. Okay. 550 is absorbed by carotenoids. Xanthophyll is absorbing some wavelength. Chlorophyll B is absorbing some wavelength. It is absorbing. Now what happens now? All the energy absorbed by the carotenoids, xanthophyll, chlorophyll B, entire energy is being transferred to chlorophyll A. Entire thing gets transferred to chlorophyll A. It happens, right? When there is one ultimate boss like Gunda for one area, he can't go every road to road to ask money from everyone. He can't do like that. So he has multiple underlings who will go do the small work. They are doing it. So all his underlings will take the energy and give it to chlorophyll A. If you see Dragon Ball Z, there's something called as spirit bomb by Goku. Okay, when this Goku is able to give, when it's going to give the spirit bomb, he will take energy from everyone. Now imagine Goku is your chlorophyll A. He's absorbing all the energy from everyone. Simple, same thing. Okay, spirit bomb. That's how I remember it also. Okay, spirit bomb. So chlorophyll B, xanthophyll, carotenoids, all of these are also called as antenna. They are called as antenna molecules. Antenna molecules. These antenna molecules are giving all the energy to the chlorophyll A, which is also called as the reaction center or chief pigment. See here. There is antenna and then there is reaction center. Reaction center is the chlorophyll A. There is the reaction center is the chlorophyll A, also called as the chief pigment, right? And this is the antenna or light harvesting antenna, which is giving all the energy to reaction center. Okay, center. Okay. Now what is happening? Understand, students. What is photosystem now? What is photosystem? Small concept, understand. Okay. First of all, understand what is light harvesting complex. This entire thing is called as LHC. Entire thing is called as light harvesting complex. A light harvesting complex has antenna molecules, it has reaction center. So LHC is made up of reaction center as well as antenna molecules. That is a LHC. This entire thing is called as a LHC. All the antennas, reaction center on top, ultimate boss on the top. Entire thing is called as an LHC. Now students, understand what is a photosystem now. What is a photosystem? A photosystem is much more complex. A photosystem has three main parts. For example, it has one part is the LHC. It has LHC, which has all the antenna as well as the reaction center. LHC is there. Okay. The other main component is the electron carriers. And the other main component is certain enzymes plus proteins. This entire thing is called as a photosystem. Entire thing is called as a photosystem. So this is the photosystem. All of you know what is LHC? This is a LHC, light harvesting complex. That is how they're harvesting the light. Light harvesting. Harvesting of light is happening. Harvesting of light is happening to the ultimate boss. Okay. Light harvesting complex and photosystem. Now students, there are two types of photosystems. Photosystem 2, photosystem 1. Okay, understand. Photosystem 2 is located in the stacked regions. Here, on the stacked regions, okay, of thylakoid membrane. Photosystem 1 and ADP synthase are found in the unstacked regions. That is the connecting link also. Connecting link protruding into the stroma. So PS1 is present in the stromal lamellae and also on the stacked regions. Remember that. So photosystem 2 and photosystem 1. Now what is the difference? What is the difference between photosystem 2 and photosystem 1? Small difference. Okay. All of you drink some water. All of you drink some water quickly. And also all of you quickly smash the like button. 150 likes is the target today. 150 likes. Okay. Now understand. Photosystem 1 works at P700. At 700 wavelength, photosystem 1 works. PS2 works at 680 nanometer. 700 nanometer and photosystem 2 works at 680 nanometer. Okay. Photosystem 1 lies on the outer surface of the thylakoid membrane, occurs on the inner surface of the thylakoid membrane. Okay. Found in both One is found in both grana and stromal lamellae. Found on the grana and stromal connecting link, stromal lamellae. While PS2 is only found where? On the stromal lamellae. That is, PS2 is found only where? If this is the stacks, here we find PS2. PS2 is found only here, not anywhere else. Not anywhere else. Okay. What is LHC? LHC is called as light harvesting complex. Light harvesting complex is made up of two main things. See this. Light harvesting complex is made up of the antenna molecules as well as the reaction center. Now, what is antenna molecule? Antenna molecules are basically chlorophyll B, carotenoids, xanthophyll. Axillary pigments also called as antenna molecules. What is reaction center? Chief pigment, chlorophyll A. Chlorophyll A. Okay. This and together they are called as light harvesting complex. Light harvesting complex. This is photosystem. This is photosystem. Okay. Now I'll tell you one more function here. Understand. First function is what? Absorption of light and transfer it to chlorophyll A. All of you understood that. First main function. Second main function is it protects the chlorophyll A from the photooxidation. What is the meaning of that? Understand. Now imagine, students, imagine if all the wavelength of light, all the way from the 400 to 700, the entire wavelength of light, if everything goes to the chlorophyll A, it will get destroyed. To prevent the destruction or photooxidation of chlorophyll A, axillary pigments will help in that. They will take some part of it. Okay, that is preventing the chlorophyll A from photooxidation, and also they help in widening the search of light. So if even though 680 is not being absorbed, all the other wavelength is absorbed and energy is transferred to chlorophyll A. Energy is transferred to the chlorophyll A. Okay. So with that, students, half the chapter is over. With that, half of the chapter is over right here. Now we come to the main part where a lot of questions are asked every single year. Many questions are asked. Can we start now, students? Can we start a beautiful concept right now called as your actual photosynthesis? Can we start now, students? The actual photosynthesis, that is, actual photosynthesis is mainly divided into two parts. Light dependent reaction, reaction where light is required. Other one is called as light independent reaction, also called as dark reaction where light is not required. Light is not required here. Okay. So this is light dependent reaction. This is called as light independent, no light, also called as dark reaction. But in light reaction, ATP and NADH, which is formed in the light reaction, it is used in the dark reaction to produce the glucose or the sugar here. Okay. So light reaction now. Light reaction also of two types. One is called as non-cyclic. One is called as cyclic. Okay. One is called as non-cyclic. One is called as cyclic. Now both of them are different. Location also is very different. Okay. Non-cyclic and cyclic photophosphorylation. Now students, let's start with non-cyclic now. Can we start? Yes. All of you open your books. And write down. Write down one more time. Ready? All of you start writing down. Now here we have the main boss. Let's take the main boss, that is the chlorophyll A. Here we have the chlorophyll A, which is the main boss. Okay. Around the main boss, do we have the antennas? Yes, we have the antennas. Okay. Now this photosystem is called as PS2. PS2 works at which range, students? Tell me. PS2 works at which range? Tell me, it works at which range? PS2 works at 680 nanometer. 680 nanometer. Okay. Now, let's take sunlight is coming. Sunlight is entering the chat now. Sunlight. Sunlight is falling on this. So all the energy is transferred. All the energy is transferred to chlorophyll A. How we learned here? Entire light harvesting complex. Entire light harvesting complex. Energy is being transferred. Energy is being transferred to chlorophyll A. Chlorophyll A. Now what happens? Understand? Obviously, it is a pigment. When energy is given to a pigment, electrons get excited. I told you in pigment. So all the electrons here get excited. Electrons get excited here. Electrons getting excited. Now these electrons are picked up by primary electron acceptor. Electrons are picked up by, first of all, it is picked up by PEA, primary electron acceptor. Can anyone tell me which is the primary electron acceptor in the case of non-cyclic? Anyone? Which is the primary electron acceptor? That is pheophytin. Pheophytin is the primary electron acceptor. Now from pheophytin, listen to me very carefully. Electrons sent to PQ, CY and PC. PQ, CY, PC. Okay. Now what is this PQ, CY, PC, students? PQ, CY, PC. Understand? This PQ is nothing but plastoquinone. PQ is called as plastoquinone. Okay. PQ is called as plastoquinone. PQ. What is CY? Understand? Very simple. What is CY, students? Does anyone know what is CY? PQ is plastoquinone. CY is cytochrome B6F complex. Now what is PC? PC is called as plastocyanin. This PC is plastocyanin. Plastocyanin. So PQC by PC. PQC by PC is the entire order in which the electrons are being transferred. The electrons are being transferred. Electrons are being transferred. And with the, whenever the electrons are being transferred, between PQ and CY, 1 ADP gets converted to 1 ATP. Convert to ATP. Okay. PQ to CY, ADP gets converted to ATP. Okay. Till here it is very clear to all of you. First is photosystem 2, 680. Electrons get excited. Pheophytin is the primary electron acceptor here. PQ, CY, PC. All the names are given there. Okay. Now understand. Now from here, the electrons are now given to the next party. Does anyone know who is the next party here, students? The next party here is very simple. That is the next boss. Again chlorophyll A. Who has a lot of antennas. Who will also take the sunlight, who will also pick up the sunlight and transfer it to chlorophyll A. But this one is called as PS2. It is called as PS2. So PS1. It's called as PS1. If PS2 is working at 680, PS1 will work at 700 nanometer. 700 nanometer. Okay, 700 nanometer. Done. Now again from here also electrons are getting excited. Electrons are getting excited here. Now from here electrons are again taken up by primary electron acceptor. Okay, let's say 2 electrons are being popped up. Okay. The primary electron acceptor is FeS, iron sulfur centers. FeS is the primary electron acceptor here. FeS. From FeS, the electron goes to FD. Electron goes to FD. Okay. From FD, now the electrons will go to the final electron acceptor. The final electron acceptor in the case of the final electron acceptor, students, listen to me very carefully now. The final electron acceptor in the case of non-cyclic photophosphorylation is none other than NAD+. It is NAD+. NAD+ will take the electrons. NAD+ will take the electrons and also at the same time it will pick up 2H+ from stroma. 2H+ from stroma is also picked up. So addition of electrons and addition of protons is called as reduction. It is called as reduction. So this NAD+ gets completely reduced to NADPH2. NADPH2. And this entire reaction is enzyme mediated, and the enzyme involved is FNR enzyme, ferredoxin NADPH reductase enzyme. Okay, and that is the entire non-cyclic photophosphorylation. I'm going to move aside, take a picture, chill. All of you, take a picture. Or if you rode with me, it's even better. That is the entire non-cyclic photophosphorylation. Beautiful it is. Entire process happening. Entire process. Non-cyclic photophosphorylation. And students, I'll make you write a few points here, But before that, students, electrons are being sent here. I'll repeat, I'll give you a small recap, understand. Chlorophyll A, PS2, 680 electrons, primary electron acceptor, PQ, C by PC, plastoquinone, cytochrome B6 of complex, plastocyanin. Then PS1, electrons, FES, FD, NAD2, NADPH2, FNR is the enzyme, electrons and protons have been added here. Now here, the electrons are being consumed continuously. Electrons are being used continuously, electrons are being consumed. But do we have infinite supply of electrons here? Where are the electrons even coming from? The electrons here are actually coming from a specific point here. Does anyone know where? In the lumen side. Here. Anyone? Where is the electrons coming from, students? Near the PS2. Near the PS2, inside the lumen part, what is happening? The water is being split. The water is being split in here. The water is splitting, that is also called as hydrolysis of water, and this hydrolysis of water is releasing 2H plus oxygen and 2 electrons. Now here, students, understand the fate of each one of them. These electrons are given here. Electrons are given here. Okay. This H2, this H plus is sent. It is sent for ATP synthesis. Remember this. We'll come back to this. We will come back to this when I'm teaching you a concept called as chemiosmotic hypothesis. This oxygen, student, this oxygen right here is released as a byproduct. is released as a byproduct. So electrons are taken up here, oxygen byproduct, H plus is sent for ATP synthesis. It is sent for ATP synthesis. This is the entire thing is called as non-cyclic photophosphorylation. Non-cyclic photophosphorylation. Now students, what did we learn here? First of all, we learned that O2 is evolved. O2 has been evolved here. Yes, O2 has been evolved. Okay. There is splitting of water. Splitting of water is also happening. There is ATP synthesis. ATP synthesis happening. It's happening. There is NADPH formation. Yes, NADPH formation is happening. This entire thing is called as non-cyclic. Entire thing is called as non-cyclic in nature. Now, apart from that, students, apart from that, if you look at the shape here, doesn't it look like Z shape? Yes. This entire thing is also called as Z scheme. It's also called as Z scheme. So this is called as your non-cyclic photophosphorylation. That is all. Boom. Over. Okay. Clear, students? Yes, Gautam, it is coming from H2O itself. Can you see here? From H2O, oxygen is coming as a byproduct. Entire thing is called as non-cyclic photophosphorylation. Beautiful. Now, if you learn this, cyclic is very easy. Cyclic is very easy. What is happening in cyclic? In case of cyclic, here we have chlorophyll A. Again, around the chlorophyll A, there is, around the reaction center, there are multiple antennas. Yes, there are multiple antennas. Then there is sunlight coming in. To chlorophyll A. All the energy is being transferred to the reaction center because they cannot do their own reaction. They have to send it to the reaction center. Okay. Now here electrons get excited here. Electrons are getting excited. Now, who is the primary electron acceptor here? Primary electron acceptor. So in the case of non-cyclic, here we only have photosystem 1, which works at 700 nanometer. 700 nanometer. Where is it present? Students, tell me. If this was the entire chloroplast, where do you find only PS1? PS1 was present, only PS1 was present on the stromal lamellae. All of you remember? Present on the stromal lamella, right? The connecting link, only in that place we had the PS1. So your entire cyclic will happen only there, not on the thylakoid. This entire reaction will not happen on thylakoids. Okay. Now here, primary electron acceptor is FES again. FES. FES is sending the electrons to FD. FD is sending to PQ again, students. PQ, CY, and PC. PQ, CY, PC. This is the entire cyclic. Now here, PQ to CY, one ATP formation from ADP. ADP formation is happening, and between FES and FD also, one more ATP formation is happening from ADP. Now, after entire reaction where electrons are being transferred, electrons are being constantly being transferred. In the case of cyclic, electrons are again sent back to the PS1 in a cyclic manner. It is again being sent back. That is why it is called as cyclic photophosphorylation. This one is called as non-cyclic photophosphorylation. Here, there is no oxygen being released. There is no ADP formation. There is no splitting of water. Nothing. Only thing which is happening here is ATP formation. Only and only ATP formation is happening here. ATP formation is happening here. That is all. That is all is happening here, students. Okay. That is all is happening in the case of cyclic photophosphorylation. So what is the difference? Small difference we'll see. Small difference we'll see now. Okay. Cyclic versus non-cyclic. In the case of cyclic, electrons move in a cyclic pattern. Cyclic pattern. Electrons in a linear pattern. They don't come back here. They don't come back. Only photosystem 1 is involved. Yes, only one is involved. Both photosystem 1 and 2 both are involved here. Both are involved. Electrons are first expelled from the reaction center 1. Yes or no? From 1 they are expelled out here. But in the case of non-cyclic, from the 2 they are expelled out. 2 they are expelled out. Okay, from 2 they are expelled out. Okay. Electrons return back to P700 after passing through ETS. Electrons are coming back. Here they are not, do not return. Synthesis of ATP during the light reaction of photosynthesis occur by cyclic passage of electrons through and from P700. Synthesis of ATP during the light reaction of photosynthesis in which an electron donor is required and oxygen is released as a byproduct. That is, electron donor is a water here. Occurs in the isolated chloroplast and photosynthetic bacteria. Occurs in higher plants and cyanobacteria. Occurs in anaxogenic photosynthesis. No oxygen being evolved here. Oxygen is being evolved here. Okay. Final electron acceptor is P700. Final electron acceptor is NAD+. Students, each and every one is actually here a PYQ. Photolysis does not occur here. No photolysis of water. Photolysis occur here in the case of non-cyclic. And students, if you want these notes, right? If you want these notes, everything will be available on Telegram channel. And also students, not only Telegram channel, it is also available on the treasure box. If you go treasure box, it is going to be there. Okay. Now, exactly. Here it follows Z scheme. No Z scheme. No Z scheme here. Okay. Z scheme is followed in the case of non-cyclic. All of you are following beautiful today. Okay. Now students, I will be teaching you rather very easy concept which sounds difficult but it is very easy. That is called as chemiosmotic hypothesis. It sounds very fancy, no? See, all of you, if you hear first time chemiosmotic hypothesis, I'm like, oh my god, such difficult thing to hear only. It is difficult? No, it is not difficult. It is very simple. It may sound difficult but it is very easy. Just pay attention for the next 5 minutes. Next 5 minutes, if you pay attention, I will teach you the chemiosmotic hypothesis. Just 5 minutes. Ready, all of you? Ready? All of you drink some water. All of you drink some water and we'll start. Okay. After this, I'll give you the cutie break. After this, I'll give you a small break and then we will start dark reaction. So we will finish light completely, then we'll go for dark. Okay. Exactly. It's very easy. Okay. Students, let me tell you again. Your plant physiology is actually very easy. If you just attend one of my lectures, you will know it. If you attend one of my lectures, I make it so detailed and precise that you cannot go wrong anywhere. Not possible only. Okay. You attend this lecture, download the notes, read NCERT once. There's a very high chance that you will not lose a single mark from this chapter. Very high chance. Okay. Now students, now here I told you, here I mentioned in the case of non-cyclic photophosphorylation, here I told you ATP is produced. Here I told you in the case of non-cyclic ATP is produced. ATP is produced. But how is it produced? How do we explain that? How do we explain the process of ATP formation? the entire process of ATP formation, how the ATP is being formed, it is explained by chemiosmotic hypothesis. So chemiosmotic hypothesis is very simple. It explains how the ATP is being produced by the plant. That is all is chemiosmotic. That is all it. That is all. If you understood that much, you're good to go. Okay. But I will add 2 more points. Listen to me. Okay. That is, see, chemiosmotic hypothesis explains the mechanism of synthesis of ATP. That is all. That is all. Okay. Now this is the entire reaction. This is the entire reaction. Now I will show you in small video or small animation. You will get a better clarity on how things happen. Ready, all of you? Ready? Let's finish this first and then I'll show you. I'll finish this and then I'll show you. You'll get a better clarity itself before I go. Okay. Now students, what is the structure first of all? What is the structure? Now if you go back to this particular animation, do you remember this? Here I told you this part. All of you can see this? This is a thylakoid. This is a thylakoid right here. This is a thylakoid right here. Can you see the inner part? Inner part is called as lumen. This inner part is called as lumen. Lumen. Okay. This entire thing is a thylakoid. Entire thing is a thylakoid, and middle part is called as the lumen. So what is the structure? The structure right here is basically, this is a grana, and this individual, if you highlight one individual thylakoid, this is the structure which you get. Okay. Now understand. What is this on top? What is on top? What are the students? PS2, PS1 for oxidation of water, PQ, CY, PC. What is this, students? What is this? What is this? Students, PQ, CY, PC. Isn't this right here non-cyclic photophosphorylation? Non-cyclic photophosphorylation right here on the thing. So if I ask you, I told you already in the starting of the video, I told you, right, on top of this thylakoid, I will find both PS2 and PS1 right here. PS2 and PS1. So where are they found? PS2 and PS1 are found on top of the thylakoid. So this right here is an entire thylakoid where non-cyclic photophosphorylation is happening on the top. It is happening on the top. That is the entire non-cyclic. Beautiful concept. Very easy to understand. Okay. Now this entire reaction takes place. If this entire reaction takes place, there are 4 requirements. There are 4 main requirements. For example, the first one is ATP synthase, let's say. ATP synthase. Second requirement, intact membrane. Intact membrane. Third requirement, proton pump. And the last requirement, which is the most important requirement, the fourth one, the most important requirement is proton or H plus gradient. Now, what is the H plus gradient, students? What is the H plus gradient? Understand. The H plus gradient, the meaning of gradient is difference. We need a difference in the H plus. Now you might ask me, difference between what? We need to make high H plus. Can you see here? High H plus inside the lumen and low H plus in the outside. Now how do we achieve that? How do we achieve high H plus inside the lumen and low H plus outside? Small 3 methods are there. 3 methods. First method, understand. First method is right here. First method. when there was photolysis of water, I told you, electron was sent here, oxygen was released outside, this hydrogen is sent here. So the first method is coming from here. So can I tell H plus concentration inside is increasing because of this now? All of you agree with me? We learned here also. See here. Electrons were given here. Oxygen was released as a byproduct. H plus was given for ATP synthesis. Same thing is happening here also. H plus is given for ATP synthesis. In the first method. Second thing, this plastoquinone is also a hydrogen donor. It will take H plus from the lumen, from the stroma and send it inside. So by that method, can I tell the H plus concentration is increasing on the inside and outside H plus concentration is decreasing because plastoquinone is taking H plus from the stroma and pulling it inside. So by this method, H plus concentration is increasing inside the lumen. Second method. This is the first method. This is the second method. Now third method is right here. In the last and final step of non-cyclic photophosphorylation, do you remember that in the last step, did we take 2H plus from the stroma for this reduction to take place? Yes, we did, from the stroma. So can I tell, because of the last step in the non-cyclic, the H plus concentration is again decreasing. In the outside. So in the stroma, H plus concentration is decreasing. Inside the lumen, H plus concentration is increasing. By a simple method of gradient formation, we can make ATP. How? Understand. Whenever 4H plus is passing through, this ATP synthase right here, the ADP gets converted to ATP. So 4H plus is giving you 1 ATP here, and that is how ATP formation takes place in the chemiosmotic hypothesis. I'll let this entire concept get settled inside your brain. Settled inside your brain right now. See, this is the chloroplast right here. This entire thing is the chloroplast. Light particles are going here. Okay. The entire light particle is taken up by the thylakoid. This entire thing is the thylakoid right here. It is taking up. But where exactly is falling, students? On thylakoid, I have told you. On the thylakoid, there is both PS2 and PS1. Both are present on the thylakoid. Yes. Both are present on the thylakoid. See here. See. This is the membrane. This is the membrane of thylakoid. We have the PS2 and PS1. This PS2 and PS1 are absorbing the light energy, and they're passing it. Can you see? Light energy is being passed to the main reaction center. Can all of you see this? This is the antenna molecules. Antenna molecules are taking the energy and sending it to the reaction center. Electrons being sent to the reaction center. This is chlorophyll A. Now from here, electrons are sent up, basically to the next electron carrier, primary electron acceptor pheophytin. Now pheophytin is transferring the electrons to the next electron carrier and finally to the photosystem 1, entire non-cyclic. From here, once enough of electrons are there, in the lumen, Once lots of electrons are there, see this is 4 lights of water in the lumen. Electrons are being constantly supplied. 4 lights of water is happening in the lumen side. In the lumen, 4 lights of water. Electrons are sent here. Oxygen is released. Protons are sent there. Being continuous process. So what happens eventually, students? Here, can you see NAD plus formation happens at the end of at the end of photosystem 1. NAD formation happens, and all the H plus is sent here, and this H plus is rotating the ATP synthesis, and ATP formation happens. This is the chemiosmotic hypothesis. That is the chemiosmotic hypothesis, and that is how ATP and NADH is produced out of light reaction. Out of light reaction, students. Out of light reaction. And that is how this entire thing works. Entire thing. Non-cyclic PS2, PS1 as well as ATP synthesis. ATP formation, NADH formation, NADPH formation here. And that is how entire light reaction takes place. Entire light reaction takes place. That is the end of beautiful concept of light reaction. Done. Done, students, done. Sir, how H plus influences pH and how pH decreases? Because obviously protons have a direct connection with the pH. Yes or no? Right. That is the entire process of light reaction. Now we'll take a small break, students. I don't have my watch today. The time is now 8:52. Let's come back at 9:10, students, or 9:15. Let's come back at 9. Hmm, 9:15 itself. Let's just take 9:15. Okay, so all of you go eat something and come back. Okay, so I'll see all of you. I will see you exactly. This is a dinner break. So all of you go eat the dinner. By 9:15 you should be back. 9:15 all of you should be back. Okay, promise me. Okay, so take care until then. So hello students, quickly let me know in the chat right now if I'm audible and visible to each and every one of you. Quickly let me know in the chat right now. Now students, let's say around 70% of the chapter is over. Mainly where we have to study about the entire light reaction, the early experiments, entire thing is over. Now we are hardly left with, let's say, around three concepts. That is, what is dark reaction, what is C3, what is C2, what is C4 cycle. Now if these three concepts are over, the entire chapter is over. Okay, so with the same energy and the Josh, let's start the dark cycle now. Let's go with the dark cycle now. Okay, the first thing which you need to understand here is, students, let's erase this part here. Okay, in the case of dark reaction, it does not require light. True or false? True or false, students? Okay, true. Yes. Students, quickly tell me in the chat. Dark reaction does not require light. True or false? Quickly. You would like true, say true. Dark reaction does not require light. Okay. One more statement. Dark reaction needs products of light reaction. True or false? Tell me. Dark reaction requires products of light reaction. That is true. That is, in the case of light reaction, we produce NADPH. We produce ATP. The ATP and NADPH is used in the dark reaction. It is used in the dark reaction. Does not require light. Also called as Calvin cycle because the scientist requires the products of light reaction, NADPH and ATP. So, in the light reaction, with the help of light energy, ATP and NADPH is made, that is transferred to the dark reaction. The entire dark reaction occurs in the stroma. The entire dark reaction occurs in the stroma. Also the matrix. It is also called as light independent reaction. Why? It is called as light independent reaction because it does not require light. Okay. Now, it is also called as PCR cycle, photosynthetic carbon reduction cycle, right? Where the product is glucose, the product here. Raw materials, ATP, NADH, CO2. Three main raw material for the dark reaction. Three main steps. Students, remember, entire Kelvin cycle, also called as C3 cycle, also called as dark reaction. The small trick here is 3-2-1 trick. There is a trick called as 3-2-1-0 trick. Now what is the trick here? 3 means 3 steps and also 3 ATP is used. 2 stands for 2 NADH is used. 1 stands for 1 CO2 and 0 stands for no energy is produced here. No energy is produced. Okay. So with that, students, let's write down the steps in the entire C3 cycle, also called as the Kelvin cycle. Kelvin cycle or C3 cycle, also called as PCR cycle. Okay. The first step is called as carboxylation. In the first step, the RUBP. Students, in the first step, RUBP. That is ribulose 1,5 bisphosphate. Ribulose 1,5 bisphosphate. Ribulose 1,5 bisphosphate is converted. 1,5 bisphosphate is converted to something called as PGA. 3-PGA. What is PGA? Phosphoglyceric acid. It is called as phosphoglycerate. Phosphoglycerate. This is the entire thing is the first step. This entire thing, the first step, the entry is happening with the help of CO2. Okay, now students, tell me, here we have 1 carbon. RUBP is 5 carbons. This is 1 carbon. How many carbon should we have here? Tell me, students. How many carbon should be here? Here we have 5 carbon, 1 carbon. So in total it should be 6 carbon. That is what even scientists were thinking about. But it is actually not the case. This PGA which is formed here is actually 3 carbon. Then sir, 5 plus 1 is 6. That is 3 here. So here what happens is 2 molecules of PGA takes place. 2 molecules of PGA. Since there are 2 molecules of PG here, 3 2s are 6 carbon. So 6 carbon here, 6 carbon here, and everything makes sense here. Everything makes sense. The first step is called as carboxylation. Carboxylation. In this entire step of carboxylation, the enzyme involved, very important. The enzyme involved in the carboxylation is called as rubisco. It is called as rubisco. Rubisco enzyme is involved here. Rubisco enzyme is involved in carboxylation. First step done. First step done. CO2 RUBP. 5 plus 1, 6 carbon. Now students, understand, all of you. Now this PGA now gets converted to a product called as triose phosphate. Triose phosphate. Triose phosphate. And this triose phosphate right here is also 3 carbon. 3 carbon. And this triose phosphate eventually gets converted to glucose. Eventually gets converted to glucose. Okay. Now students, understand. One second, let me just write this side. Let me pull this a little. It'll be in the center. Triose phosphate, 3 carbon. 3 carbon. This eventually gets converted to glucose. So that's how glucose formation occurs. Now students, understand. This second step is called as reduction. Second step is called as reduction. So what exactly is happening in this step? In this step, the PG is getting converted to triose phosphate. But is it free of cost? No, it is not free of cost at all. In this second step, reduction step, there is usage of 2 ATP and 2 NADH. There is usage of 2 ATP and 2 NADH. 2 ATP is converted to 2 ADP. 2 ADP. 2 NAD is getting converted to 2 NAD+. So in the second step, 2 ATP and 2 NADH is getting completely consumed. Now understand, all of you. This triose phosphate eventually again converts back to the RUBP, and this final step is called as regeneration. It's called as regeneration step. And in this step also 1 ATP is used. 1 ATP is used. 1 ATP is getting converted to 1 ADP. So students, in total it is called as 3, 2, 1, 0 method. There are 3 different steps: carboxylation, reduction, regeneration. 3 steps. 3. How many ATP used? 3 ATP. 2 plus 1, 3 ATP. How many NADH? 2 NADH is used here. For how many? Entire cycle is happening for 1 CO2. For 1 CO2. 1 CO2. Is there any energy being produced here? No energy is being produced here. So 0. So 3, 2, 1, 0, and this is the entire cycle called as Kelvin cycle. Now you might be wondering, why is it called as C3 cycle? Because of this. The first stable product, PGA, is 3 carbon, C3 cycle, also called as C3 cycle. Beautiful, right? Done. Now students, that was for one CO2. We learned right now for one CO2. We learned right now, students, for one CO2. For one CO2, we need to spend 3 ATP and 2 NADH. For one CO2. How many do you think we have to spend for one glucose? Tell me, students. Here we have 6 carbon. For one carbon, it is this much. So all you need to do is multiply into 6. All of you agree with me? All you need to do is into 6, 18 ATP, 12 NADH, that is all. This is for one glucose. So such questions can be asked. For one CO2 is how much? For one glucose it is how much? You should be able to answer. Okay? You should be able to answer. Same thing is given here. Here are a few examples. Wheat, soybean, oats. Examples of C3 plants. Now students, there's a process called as photorespiration. Yes, Gautam, it is called as Kelvin cycle because of Melvin Kelvin. Because of Melvin Kelvin, it is called as Kelvin cycle. Rhymes. Now, what is photorespiration? I'll give you a simple idea. Just focus it. Okay. For example, students, in photorespiration, I have an enzyme called as Rubisco enzyme. Okay. This enzyme is a little kalla here. One little fickle-minded. Okay. This Rubisco can bind with CO2 as we saw in Kelvin cycle or C3 cycle, or this Rubisco can form a bond with even oxygen also is possible. Why? Because it is called as Rubisco carboxylase oxygenase, CO, carboxylase oxygenase. So it can form a bond with oxygen, bond with CO2. Now when does it do it? Now when both are equal, Rubisco will go with CO2, nicely form a bond. But as soon as oxygen starts to go up, Rubisco will form a bond with oxygen, leave CO2, go with oxygen. Or as soon as CO2 goes down, again Rubisco goes form a bond with oxygen. So there's a very high chance that if there is more oxygen or less CO2 within a plant, RUBP will form a bond with oxygen, and that is called as photorespiration. So in photorespiration, what happens is RUBP does not form a bond with carbon dioxide, because this enzyme is working for oxygen now. Okay, so what is happening is RUBP plus oxygen is happening in the case of photorespiration. RUBP is 5 carbon, 0 carbon. So here we should have a 5 carbon molecule. So what do we get here is, we get something called as PGA, which is 3 carbon plus PG. PG is 2 carbon. 3 plus 2, 5. PGA, all of you know, phosphoglyceric acid or phosphoglycerate. Now what is PG? PG is called as phosphoglycolate. It is called as phosphoglycolate. Now this phosphoglycolate eventually gets converted back to PGA. But students, let me tell you, in this entire process is a wasteful process, complete waste body it is. You might ask me why, sir, why do we call it waste body? Why are we calling it as waste now? Students, tell me, when do you call someone waste? In my house also they tell you, in the house they tell you, waste body you are. When do you call waste body? When you're very lazy, not doing any work. Same thing is happening here. That is in photorespiration, students. In the case of photorespiration, here what is happening here is, in photorespiration, it is called as the entire thing is called as a wasteful process, because there is no sugar formation here. No sugar. At least in C3 cycle, there was glucose formation was happening. No glucose here. No glucose. Okay. No ATP synthesis. No ATP synthesis. ATP is used here. ATP is being used. And since CO2 is not being used, CO2 is evolved. So waste of CO2 is happening. Waste of CO2 is also happening. And also, main point, students, this photorespiration does not happen in C4 plants. So this photorespiration, this entire concept of photorespiration, where Rubisco is forming a bond with oxygen, that is RUBP plus oxygen, this entire thing, photorespiration, only and only happens in the C3 plants. In C3 plants. Only C3 plants. Now you might ask me, sir, why? Why is C4 so special? Are they really that smart? Yes, students. C4 are actually very, very smart they are. Let me tell you how. Let me tell you how they are smart. Understand. So before we go to why they are smart, you need to understand that when does the photorespiration occur? When does it occur? I already told you that first point is when the oxygen is more. Second point is that when CO2 is less, CO2 becomes completely less. Third point is that when there is a, let's say, high temperature and pressure, when there is high light and temperature, when there is high light and high temperature, C2 cycle occurs. In these three scenarios, the entire C2 cycle occurs. Otherwise, C2 cycle does not occur, and it only occurs in the C3 plants. Only occurs in the C3 plant. That's all. That is all. Okay. Now you might ask me, sir, what is so special about C4? Why don't photorespiration occur in C4? I'll give you a small clue. Okay, small clue. Imagine, students, you have a small kid running in your house. Because the small kid is falling and you are getting scolded for it, what do? Take that small kid and lock him in a room. Take the small kid, lock him in the room itself. No more trouble, no more trouble. Same thing is happening. Same thing is happening in the case of C4 plants. C4 plants are locking. Understand, students, understand this concept now. Understand, students, understand. Here we have Rubisco. Okay. Rubisco is trapped in a area. Rubisco is trapped in an area where we have only and only CO2. Oxygen cannot enter here. Oxygen entry is not allowed. Not happening. Oxygen is not entering. Now in the presence of Rubisco, wherever there is Rubisco, there is only CO2. Now you tell me, is there a chance? Is there a chance that photorespiration can occur now? Not possible. There is no oxygen only. There is no oxygen itself for photorespiration. Then how will it happen? It will not happen at all. So there is no photorespiration at all. Only and only CO2 is present. So no photorespiration. No photorespiration. So you might ask me, sir, how do they achieve it? How do they achieve this? See this. In the case of C4 plants, they have a special structure called as Cran's anatomy. If you see the Cran's anatomy, all the vascular bundles are completely covered by bundle sheet and mesophyll cell on the outside. Can you see? Here also. All the vascular bundles on the middle is completely covered by in a wreath-like structure. It's called as a wreath. It is called as a wreath. In a wreath-like structure, that is bundle sheets. Bundle sheets with large chloroplast and no interstellar space. When there is no interstellar space here, nothing can enter inside. Nothing can enter inside. Okay? Nothing can enter inside. Not possible to enter at all here. Okay? So that is the beauty about the Cran's anatomy. In the case of Cran's anatomy, the vascular bundles are completely covered by the bundle sheet. And bundle sheet will make sure nothing is entering inside. Anjali, can you, sir, can you about CAM plants? CAM plants is not in your syllabus, but I can teach you. It's not a big deal. CAM plant is similar to C4. CAM plant is similar to C4. The only difference is in the daytime they produce malic acid and they do the light reaction, and in the night time they do the dark reaction by converting the malic acid into the pyruvate and ATP being produced. Glucose is being produced. Okay, so they're a little sensitive. This is called as Kranz anatomy. It's called as a wreath. This is called as a wreath. Can you see here? Wreath-like arrangement. Wreath-like arrangement of vascular of the bundle sheets. Bundle sheet is a wreath-like structure here. Wreath-like arrangement. Okay. Bundle sheets are arranged in the form of wreath around the vascular bundles. Bundle sheets are characterized by having large number of chloroplasts, thick walls, and no internal space. Since this is the condition, no oxygen can enter inside, and only CO2 is there, and only normal photosynthesis will occur. No photorespiration. No photorespiration at all. Okay? No photorespiration at all. Yes, CAM pathways, desert. See, in desert adaptation, what happens is they cannot keep the stomata open all the time. Simple as it is, they need to make sure the stomata is open only in the night time. They make sure, Gautam, that the stomata is open only in the night time. So what happens is, in the night time, oxygen and CO2 is entering inside the cell. Only in the night time CO2 is entering. When CO2 is entering inside in the night time, CO2 is converted to malic acid and kept off. It is converted to malic acid and gets kept off. So in the daytime, what happens? In the daytime, when the stomata closes now, no more CO2 can enter. So in the daytime, malic acid is converted back to the RUBP, and in that process, C3 cycle takes place. Okay, in the night time. So daytime C3 cycle takes place. Nighttime nothing happens, because nighttime only exchange of gas happens because stomata is open in the night time. Okay. So C4 plants tolerate high temperature, respond to high light intensities, very good it is, lack photorespiration, greater productivity of biomass. All this is because of the special pathway. It is also called as Hatch and Slack pathway, students. It is also called as C4 pathway, also is called as Hatch and Slack pathway. Why? Because of the scientists who discovered it. Hatch and Slack. Okay. Now students, ready all of you? Ready? Ready for C4? Very simple it is. Very simple it is, students. Very simple. Looks like an hourglass, no? Looks like an hourglass. Now, in the case of C4 pathway, there are two different type of cells here. The first one is called as mesophyll cell. The second one is called as bundle sheath cell. Okay. Now what is happening? Understand, all of you. Understand. Very simple it is. Understand. C4. If you concentrate for 5 minutes, beautiful concept you'll understand today. Okay. Just concentrate. What is happening here? CO2 is entering inside. Okay. CO2 is entering. All of you understand. Just pay attention for 5 minutes. You'll take away a beautiful concept home. Now this CO2 is first picked up by not RUBP here. RUBP is not present in mesophyll. In fact, the CO2 is now picked up by something called as PEP. What is PEP, students? PEP is nothing but phosphoenol pyruvate. Phosphoenol pyruvate. Okay, phosphoenol pyruvate. This is 3 carbon. This is 1 carbon. 3 carbon plus 1 carbon, we will get a structure called as OAA. Called as OAA. Now what is OAA? Which is 4 carbon here. 4 carbon. That is why it's called as C4 cycle. 4 carbon. First stable product. The OAA is oxaloacetic acid. Oxaloacetic acid. Oxaloacetic acid. Okay. Now this OA gets converted to malic acid. Malic acid is sent into the bundle sheet. Malic acid is sent into the bundle sheet. All of you follow through. Into malic acid is sent to bundle sheet. Malic acid. Now here, this malic acid is converted back to OAA. OAA is again 4 carbon, 4 carbon. Okay, 4 carbon, 4 carbon. Okay, carbon is very important. Remember the carbon number. Now this OAA gets converted to pyruvic acid. Pyruvic acid is 3 carbon. 4 carbon, 3 carbon. So can I tell 1 carbon is lost here as CO2? 1 carbon is lost as CO2 right here. It is lost as CO2. Now students, listen to me very carefully, all of you. Inside bundle sheet, there is actually C3 cycle occurring. C3 cycle is occurring inside the bundle sheet. So C3 cycle is present in C3 plants and also in C4 plants. C3 cycle will occur in your C4 as well as C3. So what will happen here? This CO2 will be picked up by RUBP. RUBP will get converted back to PGA. PGA will get converted back to triose phosphate and glucose. This entire process will happen inside the bundle sheet. If C3 cycle is occurring here, can I tell the enzyme called as Rubisco enzyme is present here? Rubisco enzyme is present here. Now here, this particular reaction, the enzyme present here is peptidase enzyme. So they'll ask you question. Peptidase enzyme is present where? Mesophyll cell. Where is Rubisco enzyme present in C4? Bundle sheet cell. Okay. So C3 cycle occurs inside the bundle sheet cell. Okay. Occurs inside bundle sheet cell. Now this pyruvic acid is sent up. Pyruvic acid is sent up again. Pyruvic acid. Okay. Pyruvic acid is sent up. Now this pyruvic acid again gets converted back to the PEP. In this process, 2 ATP gets converted to 2 ADP. This is the entire process of C4 cycle. I'll give you a small recap one more time. Listen to me very carefully. It's very easy. CO2, first acceptor is PEP, 3 carbon. 3 plus 1, 4 carbon OA. Oxaloacetic acid, phosphoenol pyruvate. Malic acid here. OA. OA is getting converted back to pyruvic acid. Pyruvic acid is sent up. 4 carbon, 3 carbon, 1 carbon is removed. The 1 carbon is picked up by C3 cycle. C3 cycle is happening inside the C4 in the presence of Rubisco enzyme. Again sent up. Converted back to PEP. And this entire thing keeps on happening. Mesophyll cell, bundle sheet cell. Now here you need to remember what exactly happens in bundle, what exactly happens in mesophyll. Which enzyme is present here, which enzyme is present here. That is all you need to remember. That is all you need to remember right here. That's all. That is all is C4 cycle. Now students, now there is a small comparison. C3 versus C4. Let's read that and we go to the end of this chapter. Okay, let's read it. Cell type in which Calvin cycle takes place. C3 mesophyll, C4 bundle sheet. C4 bundle sheet. here. Bundle sheet, C3 cycle occurs in bundle sheet. Okay. The cell type in which initial carboxylation happens, mesophyll. Why? Because there is only one cell here. Initial fixation is happening in mesophyll here. Can you see here? Initial fixation is happening in the mesophyll. Mesophyll. How many cell types? 1, 2 cell types. Which is the primary CO2 acceptor? RUBP, PEP here. How many carbon? 5 carbon, 3 carbon. Okay. Now, primary CO2 fixation product, PGA is the first product. OA is the first product. PJ is 3 carbon, OA is 4 carbon. That is why it is called as C3 cycle, C4 cycle. C3 cycle and C4 cycle. Okay. Does the plant have rubisco? Yes, both of them have rubisco. Pepcase? No, no pepcase here, pepcase is present here. Which cell in the plant have rubisco? Mesophyll. Here, bundle sheet have rubisco. Here. Bundle sheet have rubisco. Rubisco, bundle sheet. Okay. Rubisco, bundle sheet. CO2 fixation rate under high light condition, medium. Okay. It will be very high. Doesn't matter only. High light, nothing matters here. Okay. Whether photorespiration is present at low light? No. Both low light, no photorespiration. Because low light is very easy. Nothing gets affected here. At high light, students, here photorespiration will happen big time in C3, in high light condition. Here, no photorespiration at all. Whether photorespiration would present at low CO2? No. Low CO2 concentration, it will happen here again. I told you, very high O2, very low CO2, high temperature, high light. Photorespiration starts. C3. No photorespiration. Would be present at high CO2 concentration? No. High CO2 is better. No photorespiration. Optimum pH 20 to 25 degrees Celsius, 30 to 40 degrees Celsius. Examples: wheat and rice, maize, sugarcane, sorghum. Maize, sugarcane, sorghum is the examples for C4 plants. And that is the difference between C3 and C4. So with that, students, we come to the end of this today's lecture. Last final concept before we end today's amazing class. Before that, students, I want you to get a good summary of what we learned right now. Okay. Just see this, you will be able to relate better. All of you. See this. Students, this is a typical flower. Yes or no? Typical plant is here. Typical plant. Okay. Now, if you find a leaf, okay, under the roots, what is happening? Inside the roots, all the water molecules are entering. All the water molecules are getting absorbed by the roots. Okay. Getting absorbed by the roots. Glucose, oxygen, everything is there. Okay. This is stomata. Through stomata, water is being released. And glucose is sent outside to the leaves. That is how glucose is sent bidirectional. To the leaf, ground also, up also. Okay. Both the sides is done. So that is the entire process of your, what have we learned till now? What have we learned till now is what exactly happens in photosynthesis. Glucose is produced. Glucose is sent to the shoot and the root. Bidirectional flow of glucose is happening. Now, students, the last concept is very simple. That is factors affecting photosynthesis. Yes, there are some factors. Plant will look like this otherwise. Okay. There are something called as internal factors. Number of leaf will matter. Age of leaf will matter. Orientation of leaf, if it is straight or cross. Size of the leaf, bigger the size, more photosynthesis. Orientation of chloroplast, amount of chlorophyll, orientation, number of mesophyll cell. All of these are genetic predisposition. Entire thing is made by genetics. It is fixed in every plant from the birth itself. But there are certain external factors also. Now, external factors play a very important role. Understand. Okay. These are internal factors. There are certain external factors. Okay. There are certain external factors, right, like this. There are certain external factors such as light, carbon dioxide concentration, temperature, water. Each one of them plays a role here. Each one of them plays a very crucial role. So before you go to that, there is a small law. Law of limiting factor, also called as Blackman's law. Now what does the law say that? I'll give you a simple example, understand. In your NEET preparation, for example, last year, you were preparing for NEET and you gave the NEET examination, somehow you could not clear it, some reason. Okay. Now I'll ask you, what was the reason? Did you not solve enough of PYQs? Did you not solve mock paper? Did you not revise everything? Did you skip any chapters? There will be that one factor, right? There will be that one factor which, because of that reason, because of that one reason, you could not clear NEET examination. That is holding you back. That is limiting your growth. Similarly, in the case of plant growth, there are many factors which influence the plant's growth. Many factors are there. But there is that one factor. There is that one factor which is present in least amount and not allowing the growth of the plant. That is called as limiting factor. See here, if a chemical process is affected by more than one factor, then rate is determined. Rate of photosynthesis is determined by the factor which is nearest to its minimal value. For example, in winter season, let's take winter season. In winter, sunlight is less, limiting factor. In summer season, water is less, water becomes a limiting factor. So there are many factors are there. The factor which is present in the least amount, right, that becomes a limiting factor. Summer, lesser in, limiting factor. That's all. Okay. Now, understand. Now students, all of you have seen this. Lot of sunlight. Wherever we go, in many of the parts of earth, maximum sunlight. So do you think sunlight can become a limiting factor, students? Generally speaking, sunlight is not a limiting factor. Generally speaking, sunlight cannot be a limiting factor unless it is a forest floor. At the bottom of the forest floor, where entire forest is covered by trees, the forest floor has very less light. Only in such cases, light becomes a limiting factor. Otherwise, light is not a limiting factor because plants get enough of light. A matter of fact, out of all the light, plants require only 10% of the light. Plants require only 10% of light. Okay, there are different types of light. Okay, for example, duration of light can also play a role. Short day plants, long day plants are there. Light quality can play a role. Red light and blue light are better than other lights. But what really matters is the light intensity. Light intensity, how much of light intensity you're adding. For example, if you see here, from A to B, now the restaurant A to B. In A to B, as the light is increasing, even the photosynthesis is also increasing. Photosynthesis is also increasing. But after B to C, there is a saturation. So it tells you that light really is not a limiting factor. Only up to a certain limit. Only up to a certain limit, light can be a limiting factor. But after that, no more. It is saturation. This saturation, no more it is a limiting factor. Okay, light intensity. Next is carbon dioxide. Students, understand. Carbon dioxide is actually one of the major limiting factor. How? Because CO2 is present very less in the atmosphere, very less CO2. See this. Only, right, very less CO2 is present in the atmosphere, very less CO2. That is why, students, that is why around, students, let's say around 0.3, that's all, very less CO2. That's all CO2 is present. That is, even if you increase to 0.05, complete CO2 fixation will change. Complete CO2 fixation will change if even there is a small change in CO2. Okay, small change in CO2. Now, at low intensities, neither C3 and C4 plants are affected to high CO2 condition. But at high light intensities, light is very high, both C3 and C4 plants show increased rate of photosynthesis when there is high CO2 and high light conditions. Now here, C3 and C4 will act differently. As a matter of fact, C3 plants can take more CO2. That is, C3 plants can take more CO2 and produce more sugar compared to C4. The saturation of C4 is just 360 microliter per, right? Compared to C4, it is around 450. So C3 plants can absorb more CO2, and that is why student, that is why in the greenhouse, we generally grow C3 plants. Okay, C3 plants. So C3 plants are generally used in the greenhouse. Why? Because C3 plants can show higher saturation. They can absorb more and more CO2. C3 plants. So CO2 is a good limiting factor. Okay. Temperature, student. Temperature, obviously all of you know, it is a good limiting factor. Without temperature, no enzymes will work. Low temperature, enzymes freeze. High temperature, enzymes will denature. Right? Temperature controls dark reaction as they are enzymatic. So C4 plants are more affected. C4 plants respond to high temperature and show increased rate of photosynthesis. C3 plants have low temperature at optimum. All of you know. In low, if C3 plants, we give them more temperature, they'll die. They cannot take more temperature, C3 plants. Tropical plants have higher temperature optimum than temperate forest. Last one is called as water. How does water play a role? Very simple, indirect role. Water has a indirect role. How? Understand. For example, students, this is the leaf. Here we have stomata. Stomata is there. If there is water is low, if water becomes low, stomata will close. Stomata will close. Stomata will close. If stomata closes, what happens? No CO2 entry. CO2 is not entering inside. If CO2 is not entering inside, obviously photosynthesis will decrease again. So less water, less photosynthesis. One more example I'll give you. See, this is a leaf here. Now imagine there is no water. If water is decreasing completely, if there is no water at all, what will happen? The leaf will curl inside. Leaves start curling. If leaf starts curling inwards, can I say the surface area is decreasing? Yes. Surface area is decreasing. If surface area start decreasing, can I tell? Because less area for light to fall now. So can I tell? Photosynthesis is decreasing. Photosynthesis is decreasing. So water plays a much more indirect role. In the limiting factor, as a limiting factor. And with that, students, your entire chapter of photosynthesis in higher plants is done and dusted. One short lecture. Now students, if you want the notes, let me show you. If you go here, if you go right here, let's just refresh it. All of you please make it 200. Make it 200 right now, because very good demo we have done today. Okay. If you go here right now, students, get complete NEET preparation treasure box. Click on this. Do you see here? Every single chapter NEET notes. Do you see notes here? Every single chapter notes are given here. So as soon as this chapter gets over, here I will add photosynthesis in higher plants 3D, and notes will be given here itself. So all you need to do is click on the notes, open it, add the phone number or email ID, and download the notes for free of cost. Notes are available to treasure box. Every single chapter is given here. And with that, students, that is a wrap today. Okay. So thank you so much for today's class. It was really fun. Let me know in the comment section of today's video, how did you enjoy today's class? Do you want more 3D classes? If yes, let me know which chapter you need next and I'll make it happen. So with that being said, students, thank you so much, and I will see you in the next lecture. Until then, I want everyone to keep studying, keep revising NCERT. I'm telling you, all of you. I can give NEET exam for you, and no. Why will I give the NEET exam for you? Okay. So students, go down to the comment section. Just say photosynthesis done and dusted. All of you. Photosynthesis done and dusted today. Very easy. It was very easy. Let me know in the comment section. Do not forget to feed. Let me know in the comment section which is the next chapter which you need. Okay, I'll make it happen. 3D one-shot lecture, and this is going to be the long way. It will take a long way, and one by one we'll get to the every single chapter 3D. Okay, so I hope with that we'll close today. So bye bye students. Take care. Good night.